Hello everyone, I welcome all of you to my channel Teacher Dilse. So a quick recap we will do. In the previous class we have seen what is reproduction and what is lifespan, and about asexual reproduction, that morphologically and genetically the organisms are similar and variation that is produced is minimum. Okay, now moving on with the types of asexual reproduction. Now, here I have listed down already for you the types of asexual reproduction, as you can see here, and it has got what? Has got as you can see, it has got fission, okay, fragmentation, budding, vegetative propagation, regeneration, and spore formation. So one by one we will be looking at them. So first we are going to start with fission. Now fission is of various types. So what is that? So let's see first binary fission. Then comes your multiple fission. Then comes your, there is a new term, plasmotomy. Then third one, that is your transverse fission. Or you might have heard with another name, strobilization. Okay. Now, first we are going to start with our binary fission. Okay. So, let's start with binary fission. So, in case of binary fission, see, whatever reproduction we are going to talk about, first thing which is of most important, and for binary fission also, in any reproductive division it happens, that is your replication of DNA. So what is happening here? Replication means what? Copying of DNA. So copying of DNA. DNA is being copied. Okay. Now then only, so when you have something, then only you can give to somebody else if you have an extra copy of it. Okay. Then replication, after that what is happening is your growth of cells. Growth of cells takes place. Then the third that takes place is your uh, nuclear division, or you can say karyokinesis. Okay. And fourth is your cytoplasmic division, or you can say cytokinesis. So this is called, a nuclear division is known as karyokinesis. Okay. And the another one is your cytoplasmic division, which is known as your um, cytokinesis. Okay. So now what happens in this, just I will show you. See here, if I make a cell, okay, there is a nucleus inside it. What will happen? The DNA, the cell content will replicate and it will form. Now the nucleus will start. Now it is divided, and then the two cells are formed by cytokinesis. So this is happening in your binary fission. Now in case of your multiple fission, what is happening? You can see here. See, this is your cell, and in case of binary fission, the best example. What is the best example you can give here is your amoeba and your bacterial cell. Okay. Now moving on to multiple fission. Here in this case, what do you see? There is a nucleus, and then what happens around this cell? Okay, around this cell, a coat is formed. Okay, and this coat is known as cyst. Okay, now this cell undergoes, the nucleus undergoes now division. It divides, it keeps on dividing. Okay, and this is your cyst. Then what happens? You will find now that the cytoplasm surrounds and it starts forming the membrane. Okay, so small cells are formed inside it. Okay, and these cells are released. See, when the cyst is what? The cyst is a protective coat. The cyst is your protective coat. Okay, so under unfavorable conditions, this protective coat is being formed, and whenever the conditions come favorable, the cyst will break and it will release the cells, and the cells will come out. And the best example for this case is your plasmodium. This causes your malaria. This is your plasmodium. Now, the next one that I have listed for you is your plasmotomy. So, let's look at your plasmotomy. This is the first case, this is the second case, and now the third case, that is your plasmotomy. Now, in this case, what is happening? You know, here in earlier, what was happening first? First was taking place was your now nuclear division was taking place, and this plasmotomy, it happens in those cells which are multinucleated. Okay, one of the best example is your now opalina and plasmodium. Okay, these are the best example for this. So what happens, they divide first. Here cytokinesis takes place. So first they divide into two cells. Okay, they will divide into two cells, and then they will form what? They will form many nucleus. Okay, so this is what is happening in the case of plasmotomy. Now moving on to the case of your transverse fission, or which we call as strobilization. Now as you can see here in the diagram, okay, see this diagram. This is the diagram of what? This is an example. The best example in this case is your Hydra, and the best example in this case is your tapeworm. Okay, see, in case of them, see what is happening with the worms and all. It's a means for increasing their number. So they are parasite. They want to increase in a very short time. So it's a quick means of increasing their number. So in them, what do you see here? As you can see here, now the hydra is breaking into, now transversely it is breaking. Okay, and it is forming and fission, and it is giving rise to what? It is giving rise, this is your polyp stage. Okay, and this is your medusa stage. You have read in class 11. Now this is free living, this is attached to the substratum. As you can see, now this medusa will give rise to gametes, your male and female gametes. They will fuse and they will form this polyp. So this is alternation of generation, which is seen in case of your these nidarians. Okay, so this is how they change. So this is your called as what? Strobilization. We can say it as strobilization, or we also call it as transverse fission. Okay, now moving on, a huge number of offsprings they give rise to. Okay, now moving on further to regeneration. Now there are two means. Now we have seen earlier regeneration and fragmentation. See, they both seem to be very confusing, isn't it? Because in fragmentation, what is happening? In fragmentation, what is happening? Your cell is breaking down, it's fragmenting it into pieces, and then suppose I make a plain area for you people. Okay, now if I divide this organism into parts, so you know that each part is capable of giving rise to an individual organism, and this is fragmentation. Even your fungi, the best examples of fragmentation are what? Your fungi, they divide fragmentally, isn't it? Your Spirogyra, these are all. So now you can say that now regeneration is also a kind of fragmentation, or you can say under regeneration comes your fragmentation. Why? Because regeneration is of two types. Basically, regeneration is what? Uh, it's basically it's a reverting the embryonic phase. Okay, like the developmental, the missing part of the body is again now growing in this case. So here regeneration, you can say it is of two types, that is complete regeneration, and the another is your incomplete regeneration. Okay, so in case of your this now regeneration is what? It's the reactivation, rather we can say it's the reactivation of post embryonic life, and it is what? It is the regeneration is what? Is the reactivation. You can say it is what? It is the reactivation of post embryonic life. For what? For restoring missing tissues, or lost body parts, we can say. So it is of two types, as I said, complete regeneration and incomplete regeneration. One of the best example of complete regeneration is what? Your sponges, even if you break them into very smaller pieces, each piece is able to grow into a complete organism. So what do we find? This is a case of complete regeneration. Entire body is being formed. And another case is your what? It's your case of incomplete regeneration. Like in case, one of the best example which you can give is your starfish, where from escaping from the predators, whether it is lizard or starfish, when they are escaping from the predators, what they do? They leave their tail, like lizard leaves their tail, starfish, what they do? They are able to leave their arm and go, isn't it? So, and that lost arm is able to regenerate. Okay, so this is called as incomplete regeneration. And with regeneration, what do we see? We see that as we move from lower organisms, I hope this term is clear, to higher, what do we find? That this capacity of regeneration decreases. They have high, they have high regeneration capacity. Okay, and here as you move from lower to higher organism, it decreases. Okay, and in case of humans, if I in case of vertebrates, if I talk, if I first talk about vertebrates, in vertebrates you will find there is only one group, that is Urodal, now which is in which comes your salamanders. Okay, they are able to regenerate. They have high regeneration capacity. Okay, and in humans, now your organ like liver, it can regenerate to some extent. Okay, now moving on to vegetative propagation. Now what is there are two kind of cells in our body. What are they? They are your reproductive, isn't it? They are your reproductive and non-reproductive. There are two kinds of cells, isn't it? दो ही तरह के होते हैं, non-reproductive. Now reproductive, if I'm talking about plants, okay, this reproductive part is what? Your flower. And non-reproductive part is your what? Your roots, your stems, your leaves. They are all your what? They all are your non-reproductive part, the roots, stems and leaves. Okay, now you know about, you have read in class 11th about roots, that roots have got, they also at many places have a storage row. And like in case of Jahelia, if you look at them, asparagus, है ना, there, if you see their roots, you can use their roots for propagation. Now wherever you look at, whether it is a stem, leaves, wherever, which cell is acting as a, that is the basic part which you need to understand, which is acting as what? Giving rise to a new, now complete new plant. In vegetative propagation, what is we are doing? We are taking a part of a plant, growing it, sowing it separately, isn't it? Planting it separately, and that a new plant it is giving rise to, isn't it? So in that, what is there in that part? If I take, suppose if I take a stem of a sugar cane, you know about that, there are nodes, if I take the internodal part and sow it, will it grow? No. But if I take this and I take the nodal region, then it will grow. It will grow. Why? Why will it grow? Why will not it grow in this case? Both are the parts of the stem. But why will it grow? Because you have read in class earlier, even in class 11th, you have read that this part, that this nodal region is containing what? It is containing your meristematic cells. It is containing your meristematic cells, and these meristematic cells are nothing but your totipotent cells, or you can say totipotent cells are what? They are your undifferentiated cells. They are your undifferentiated cells. What do we mean by saying undifferentiated cells? Like you are formed from a single cell zygote. That zygote was having the capacity to form what? All different kind of cells, isn't it? So that is your undifferentiated cell. Okay. So like an analogy I can give you. You have got flour, and you need it, okay, and that flour can be made into a roti, it can be made into a puri, and then a variety of dishes you can convert it, isn't it, from the same flour, isn't it? Same way, these undifferentiated cells have the capacity to give rise to various other kind of cells of your body. Okay, in regeneration also this is happening. Your undifferentiated cells, they are getting your, now in case of your regeneration, there was something else also happening. There was your, there what was happening, your differentiated cells was there, and suppose now like porifera or any other organism, you have breakdown into pieces, and those pieces are having certain cells. So those cells are what? Differentiated cells. Now they change into what? These differentiated cells, they change into these undifferentiated cells. Okay, and this process is called as de-differentiation. We call it as de-differentiation. Okay, and now these undifferentiated cells can give rise to the entire organism. So that was happening in regeneration. But here what is happening? Here in them already these regions have got what? Undifferentiated cells, and therefore when you sow them, they give rise to the complete or plant, isn't it? So this is what is happening, whether it is the case of your stems, roots, leaves, everywhere you are finding what? That these are having your undifferentiated cells at that region. Okay, So that's why it becomes easier. Now if you take this example I'm taking, um, like in case of your stems, your from your tubers till your suckers, they are what? They are the underground parts. They are what? They are the underground. Huh? They are the underground parts, isn't it? They are found under the ground, these stems. And these stems, if suppose I'm taking the example of potato, they have got what? These are the nodes. All of you have seen potato, all of you are fond of potato. If you take this part, now having these nodes, now then only it can give rise to the plant. Okay, if I take this part which is not having the node and I sow it, it won't grow. Why? Because it has got adventitious buds and they have got meristematic cells, okay, which can give rise to your entire plant. Okay, so this is, and they are found under your ground. So these are the examples: potato, bulbs के लिए garlic, onion, rhizome ginger, turmeric, okay, palms colocasia. So you know it, suckers mint, okay, runners stolon. These are what? They are your uh, sub-aerial. You can say they are sub-aerial. Now in case of runners, what is happening? Suppose this is your ground, okay, so they are just running above your ground horizontally and they give rise at the nodes regional roots and then the plant comes out. Okay, so this is the means of this propagation. Okay, so you must have seen them in oxalis. Okay, now in case of stolons, what is the difference in between in the runner and a stolon is that, see, this is the ground, okay, and in them these roots are not, they are these stems are arched. Okay, these stems are arched, so they can pass over any obstacle is there. Okay, and at these regions they can give rise to your plant. Okay, and remember that must be a nodal region. Okay, they must have a meristematic cells. So in case of leaves, the best example is your bryophyllum, as you know. And in case of bryophyllum, what do you find? That here at the notches they have got these axillary buds which separates off and it gives rise to the complete plant. And the bulbils are also your fleshy buds, okay, found in agave, lily. And your torions are what? Your fleshy buds found in aquatic plants. Like your offsets are nothing but your a type of runners which is found in your aquatic plants. Okay, so offsets are also aquatic, like in pistia and cornea they are found. Okay, so these are the examples. You have to be thorough with the examples. Okay, now so let's look at spore formation. Now in spore formation you can see that here I have taken this picture from your NCRT, as NCRT is very important. I told you people already, and therefore I am saying you that since this is an example of what? Chlamydomonas, and you look at the zoospores are motile spores and they have got flagellum on them, and they are what? A kind of an asexual spores. Okay, and here is also what you can see conidia in case of fungi, where they are, these are single spores present. And remember, in case of fungi, whenever there is an asexual spore is formed, what is happening? They have got an horizontal hyphae running, and one of the hyphae goes vertically upwards and it bears the sporangium, and the cells here divide and gives rise to spores. So these are your asexual spores. Okay, so similarly here also you can see in this diagram that you have these conidia are born vertically. You can see the hyphae is growing upward and it is having these, they are dividing and giving rise to your uh, spores. And remember one thing, that the spores are always haploid. They are always haploid. Okay, so they are always haploid, one thing to note that. And now the last one is your budding. So coming to with the budding, what we see here in this picture, you can see that this is your NCRT picture only, which I am showing you. This is your bud, isn't it? This is your exogenous bud that is found outside the body. As the example is your hydra. You can see here, even your yeast, this is an example of your budding. But here you can see that in case of your poriferans, what is happening, like your sponges and all, what they do, they form gemmules. As you can see here, the word is written gemmule, and these gemmules are what? They are a kind of an endogenous bud. They are your endogenous bud, and they uh, these archaeocytes, they come out of it and they grow into the organism. Now let's uh, look at the advantages. What are the advantages? Let's look at the advantages of asexual reproduction. So we have seen that asexual reproduction, the advantage first is that they are identical to the parent. So if the parent is very juicy, and it's having good quality, sweet, also all the quality I wanted to continue it, then I will use what? This asexual means of reproduction, and they are what? Easy to grow, and they are also what? They are easy to grow. They are quick to grow. You can grow them very quickly and economical. They are economical. Okay, this is their advantages. And even now, if environmental condition is unfavorable, then also you can grow them. Okay, so you can grow them in unfavorable conditions also. Okay, you can grow them. And what are the disadvantages? Let's look at some of the disadvantages. Okay, so what is the disadvantage? What is the disadvantage? The disadvantage is that it doesn't leads to any variation. It doesn't leads to variation, and it is very susceptible to diseases. Very easily it can catch diseases. Okay, so and the one thing more is that they are short lived. They are short lived. Okay, and these are the disadvantages. Okay, with this we have completed the topic. So thank you. In the next class we will meet with the sexual reproduction. May God bless you all.